And so now let's get to our speaker. So our speaker tonight is, as you know, Dr. Josh Bonde, and he is the research director, right?
Right.
At the Nevada Science Center, and which is supports science education and research across Nevada and the West. So, Josh, we're hoping that you'll fill us in a little bit more on your background and also about the Nevada Science Center. That would be cool if you'd give a couple of minutes to that.
Right,
So
that'd be my pleasure. Okay, let's see. Let me fire up my share screen. All right. All right, can you see my screen?
Yes.
Yes, okay.
Yes.
All right, let's go ahead and get going then. And if there are any technical issues, somebody just chime in. I have no problem being interrupted. It doesn't bother me one bit. I was a university professor for about 8 years, so I'm used to the most obnoxious of obnoxious. So there's nothing you're going to do that's going to offend me. All right, so thank you guys for having me tonight. I'm, again, Dr. Josh Bonde. I'm the Director of Research here at the Nevada Science Center. And the Nevada Science Center, like you mentioned, we're a nonprofit group that's based out of here in Southern Nevada, but we offer free virtual programming. We do like virtual field trips for students, which has been really helpful, especially over this past year. We've been able to reach students in India, Australia, across Europe, and we've seen about 100,000 students just in the last year through all the COVID stuff. So if you are an educator or know anybody that's an educator, shoot me an email after the presentation, and I'm happy to get you the information on how to get hooked up with our free programming. We also have a lab here in Henderson, Nevada. You can see some of the stuff. Well, when I stop sharing my screen, I'll show you some of the stuff that's in the lab too. But with that, a little bit about myself. I'm a born and raised Nevadan. I grew up in Fallon, Nevada, which is about an hour east of Reno. I did my bachelor's at the University of Nevada, Reno. I did my master's at Montana State University in Bozeman, and then I did my PhD back here in Nevada. And I did a stint as a university professor for a number of years. I was the curator at a local museum, and then me and my fiance decided to start our own science center just to set out on our own. So it's been scary. COVID's been not all that helpful, but hey, we're trucking on. All right, so unless there's any questions before we get going, I will start with the talk. Anybody got anything? All right, so let's go ahead and get into it. So I am going to be talking tonight about a research project that I started as part of my PhD program. I won't say how long ago that was. I don't want to date myself, but this is a project that came to light as a result of working with another PhD student that I worked with at university. So this is a reevaluation of Nevada's first frog, Eorubeta nevadensis, and the utility of fossils in paleogeographic reconstructions. I know that seems like a long, obnoxious title, and it is, but we'll get into it. It's really not that complicated. As many of you know, because you're science fans, or else you wouldn't be tuned in tonight, we like to use lots of complicated, obnoxious terms for simple terms, or for simple items, simply for job security. All right, so collaborators. So you can't do projects like this by yourself. So some of our collaborators, this is Amy Henrique from the Carnegie Museum of Natural History. She's our frog expert. Peter Druschka, who used to be at ExxonMobil, but is now a research associate here at the Nevada Science Center. And then my undergrad mentor and dear friend, Dick Hilton at Sierra College, or formerly of Sierra College. He just recently retired this past year, which is a community college over in the Sacramento area. All right, so who is Nevada's first frog? Who is Eorubeta nevadensis? This is the very first specimen that was ever found. Now, notice there's kind of this weird circular cut on this rock. That's because the specimen was actually found in an oil well drill core. And they found not just one, but they found two in this one core. So if you imagine a drill core, this one is 4 inches wide, hit 2 frogs. And so Hecht, who described this species in 1960, noted there must be a prolific number of specimens in the subsurface if one 4-inch drill core hit 2. And so he named the specimen Eorubeta nevadensis. It came from the Sheep Pass Formation, which we'll talk more about here shortly. And the Sheep Pass Formation has traditionally been dated between Cretaceous to Eocene in age. So who cares about this frog? Well, it turns out that this little frog, once Heck described it, has caused fits for frog experts. Looking at the anatomy of the bones, looking at how it all fits together time-wise, it's really difficult to figure out who this frog is related to. So who is this little frog? Who's it related to? Where was it living? And what does this mean in the grand scheme of things? So with new specimens that we're discovering as part of this research project, we can get a better sense of who this frog is actually related to. The stratigraphy, I mentioned that this stratigraphic unit, this geologic formation, is Cretaceous to Paleogene in age, which means that this frog is a survivor taxon. So we find it below and above the KP boundary. So it's definitely part, if it didn't actually span the Cretaceous Paleogene boundary, and we have not precisely pinned down where that's at in the section, it was definitely a part of the recovery fauna. So meteorite hits in Mexico, you think of all the horrible things that happened in the coastal plain. You think of all the other Laramidia stories that come to an end when the meteor hits. Up in the highlands of Nevada, this little frog keeps hopping on. And it turns out, based upon a study that Amy did, that I was a co-author on in 2018, the reference is down here at the bottom of the screen. It turns out this frog is a really primitive frog. And so it's the sister group to most living frog groups. So it doesn't fit nicely within any living frog group. So it's some sort of weird little mountain species. Mountain evolution does weird mountain things. And so we have this weird little snapshot of a primitive group hanging on someplace where, most other frogs of this variety have already passed away. So let's set the scene geologically. So because I assume there's a couple at least geo nerds in here also, because I'm a geo nerd and I love talking a little bit of geology. So we'll talk a little bit of structure and tectonics, and I'll throw some pretty pictures in there too. If you couldn't care less about rocks, you can zone out for, I'll say, 2 to 3 minutes, and I'll tell you when you can zone back in. So the late Jurassic, the west coast of North America is dominated by a big subduction zone, kind of like the west coast of South America is today. So this is the processes which create the Sierra Nevada arc. So the Sierra Nevadas were more like the Andes during this period of time. It initiates a great big series of thrust faults called the severe orogeny across the western margin of North America. So if you think of this big oceanic plate in North America as two cars hitting on the interstate, even though North America wins this fight, the fender still gets crumpled up. And so the severe orogeny is that crumpled up crust, which creates this mountain building event where Nevada is today. So while scenes like this on the right are playing out in Utah, allosaurs and sauropods and stuff, in Nevada, we're up in the mountains now. So this is, every talk has to have a Ron Blakey image. And so here's our Ron Blakey image showing this period of time from the Cretaceous to the Paleogene. Here's our Western Interior Seaway. And out here in Utah, we're on the coastal plain where you find all your cool dinosaurs. Here in Nevada, we're up in the crumpled up zone from this big impact off the west coast. So it's really hard to preserve sediments in high elevation environments through geologic time. Why is that? When you elevate something into high elevations, it's exposed to more intense weathering. It's farther away from the sea where all sediment is ultimately trying to go. And so geologically, it's really hard to preserve ancient high elevation rocks. And so what our system here is showing is that we're shedding all of our Nevada rocks over into Utah to preserve your dinosaurs. But don't worry, I'm not bitter about it. So some cool geology diagrams. Again, if you're not into geology, you can stay tuned out for a few more minutes. So here we have our subduction zone off the west coast of North America, created in the gray, this big severe orogeny belt. And so Nevada is smack dab in the middle of this. We'll talk more about some of these terms as we get into talking about more of the specifics where we're finding our frogs. So most of Nevada during this period of time is interpreted to be a high elevation plateau. Modern geological analog or rough analog would be the modern Altiplano or the Puna Altiplano of South America, where it's really high elevation. Think of places like Bolivia, portions of Argentina and Chile, Peru. that's kind of the elevations we're talking about for Nevada. And so geologists have, or at least geologists in the Great Basin, have termed ancient Nevada the Nevadenplano as kind of the as an ancient analog for the modern Altiplano. Very southern Nevada is the exception to that. So where we're at here in southern Nevada is the one part of the state that doesn't seem to have been uplifted. During this period of time, you guys out in Utah, you're out in the Foreland Basin, which is structurally called, where you get all of our sediment from Nevada is being dumped to preserve your fossils nice and neat. So again, here's our Ron Blakey map compared to the modern Altiplano Puna of South America, where this high elevation orogenic plateau, which is the result of a big subduction zone off the west coast of South America. So the same story is presumably playing out off the west coast of North America during late Cretaceous time. So what is the Sheep Pass Formation where we're getting these froggies from? And we'll get to the frogs here in a minute. So it's a big sedimentary package from these high elevation rocks, or this high elevation setting. So this is the focus of my collaborator, Peter Druska's PhD. So when I first started at UNLV, Peter was just wrapping up his PhD. And so for those of you who've been through graduate school or have been through any other hierarchical situation in life, and you're on your way out the door and the newbies coming in the other door, and you don't even really want to get to know the new people, Peter wasn't one of those types. So he's getting ready to head out the door, I'm coming in, and Peter really embraced me into the program. He said, you're one of those paleontologists, right? I said, kind of hesitantly said, yeah, because in geology programs, you never know how that is going to be responded to. And Peter said, well, I got a really cool bird fossil you have to come check out. And so I go into his office and check out his bird fossil. Well, it turns out his bird fossil wasn't a bird at all. It was a frog. And so that's how this whole project started. Peter was out there measuring stratigraphic sections and found the first frogs in outcrop from this formation. The first frogs since those drill cores that Heck described in 1960. Now, the Sheep Pass Formation is long known to be fossiliferous. So Heck described those frogs from the drill core. Steve Good described snails and clams that had been found in there. Fausch described pollen and plant fossils that have been found. And to the north, closer to the city, or I won't call it a city, the town of Ely, Embry and his collaborators discovered a bunch of mammals, which are a little bit younger than the rocks we're talking about here today. So recent age control that Peter did in 2010, I can't believe that's over a decade ago now, places the base of the formation as Late Cretaceous. So for those of you who are follow the geologic time periods of Laramidia, the very base of the Sheep Pass Formation is Campanian in age. So roughly the same age as some of those cool dinosaurs that are coming out of places like Grand Staircase. We get tons of invertebrate fossils. You can't pick up a rock out of this formation without having a clam, an ostracod, a snail, or something in it. The weird thing about this formation is vertebrates are rare and are extremely low diversity. So even though we have a sedimentary package that spans over 10 million years of geologic time, the only vertebrates that we have out of the entire dang thing are frogs. We've looked, all we have are frogs. So geologically, here we are. So this is the town of Ely. The Utah state line is over here just off the map a little bit. So I'll just do a land acknowledgement real quick since we have it on the screen. So I'm a member of the Western Shoshone, and so this is our ancestral lands, so the Newe. So just to give a shout out of respect to the living elders and past elders that called this place home and lived off of the land. So it turns out that my grandfather, talking to my aunt, actually used to drive cattle through the exact same canyon where we found these frogs. And my grandfather passed away when my mom was little, so I never actually knew him, but it's kind of cool to think about that connection to a place where we were actually probably camping in the same camps, and I probably saw some of his pitched sardine cans on the trail. So just kind of a cool personal story. But here we are in the Sheep Pass type section, just south of US 6, just west of Ely, down here in the Egan Range. So this is a stratigraphic section that Peter put together as part of his dissertation, and I threw in the froggies. And so the base is this great big megabreccia, again, dating back to Campanian time. We do have a non-conventional uranium lead carbonate date on the microbialites at about right at the very end of the Maastrichtian, right at the end of the Cretaceous here at the base of member B. We have frogs all the way through member B, and then they're found at the top of member C. And the cool thing about them being found at the top of member C is that we're talking about frog bone beds. So, that's not a term you typically hear about, but we actually have bone beds of frogs. So here we have, this is what the outcrop looks like. So this is member B, kind of the grayer beds. Member C is more of the tan colored beds. Member B is interpreted to be kind of shallow with emergent vegetation, alkaline lake. Member C is more where we have some oncolites. We have kind of delta facies building out into the shallow lake. For those of you who might know some of the folks that are in this image, this is Tina Campbell down here in the bottom center. For those of you who know, this is back in 2009. And then this gentleman over here digging into his can of cold SpaghettiOs on the outcrop, that's Tyler Berthusel over there. So some of you might know him since he's up in Salt Lake now. He was out here on the outcrop helping us dig up frogs over a decade ago. All right, so the unit B. So again, this is interpreted to be a lake, a carbonate lake environment, kind of like modern lakes in the Great Basin. It's dominated by microbialites. So we got lots of microbial fabrics, so kind of that crinkly fabric. Laminated mudstones would tell us this is kind of a calm lake. We do get fossilized earthquakes in the form of seismites. So all of a sudden you see a bunch of soft water deformation and these big slump folds. Those are indicative of earthquakes in the lake environment. And then tempestites, which is, again, the geologically obnoxious term for a storm deposit. So we can see where storms came through and ripped up mud and ripped up fossils and things like that. And the fauna that lives in Unit B includes all of our frogs. We get clams, we get snails, and we get ostracods or crustaceans. And we do get some flora as well. So on to the frogs. So we're talking taphonomy, and I'm sure many of you know what taphonomy is, but if you have missed the train, taphonomy is like the CSI, to use the Las Vegas analogy, the CSI version of paleontology. So we're going to be talking about how these frogs can be preserved where they were preserved. And so here we have this frog. This is the head. This is the shoulders, an arm, the hips, and then these are the legs going into the matrix. This crinkly fabric, this is a microbialite. And so this is where a pond scum, for lack of a better term, was during the Cretaceous and early Paleogene. But this frog died and settled to the bottom of the lake to be entombed within this microbial mat. There's actually some recent taphonomic studies that shows these microbial mats in modern settings like encapsulating frogs, and they'll build a little sarcophagus around the dead frog, which actually protects them from the surrounding environment, which is kind of an interesting story. All right, so the frogs, presumably in this taphonomic mode 1, were preserved in microbial mats. Probably these weird microbial sarcophaguses were inhibiting scavenging. So even though these frogs are sitting out there and there's lots of invertebrate trace fossils through this unit, you can see a little trace fossil through this microbialite. It means there was invertebrates there, but for some reason the invertebrates were not eating them. Even the finger bones are where they're supposed to be, and finger bones on frogs are teeny tiny. So we think the frogs likely accumulated attritionally, which just means they died through time. It was a lake, frogs live, frogs die, they settle to the bottom of the lake, they get preserved in these microbial mats to be eventually entombed in carbonate. These microbial lights are interbedded with other lithofacies, a couple of which we'll talk about. They're found in life position, and it doesn't appear that they've been transported much, if at all. On to our second way our frogs have been preserved. And so these are frogs preserved in mudstones. So unlike the pretty frog that was preserved in the microbialite, these frogs are usually found slightly askew. Usually we have a couple of elements that have been dragged off or are not where they're supposed to be. There's no evidence of microbial fabrics in these facies, but we can see that they're nearly articulated. They're a little askew. We see toe bones, a couple of finger bones. The jawbone's been disarticulated here. The left arm has been drug off somewhere. So it's a little bit different type of preservation. This is another one of these frogs. I should also mention here, we have you can go your entire paleontological career and not find a frog. In this formation, we have dozens and dozens and dozens of complete frogs. It's completely bizarre. Now, in this other frog, this is a part and counterpart. All of these little brown things, these little brown dots in the matrix, those are all ostracods. And the ostracods are actually found in ostracod to bone association. And that's kind of a sign that some ostracod species are actually scavengers. And so these frogs actually appear to have been somewhat scavenged by invertebrates. So what's the difference between frogs in mode 1 and mode 2? The frogs preserved in these laminated mudstones. They accumulated, again, attritionally through time, just like the first one. So frogs living, dying in a pond. These ones just didn't land on microbial mats. There is evidence of invertebrate scavenging. So we have ostracod swarms. We have a random distribution of disarticulated elements. So snails can do this. Crustaceans can do this. And this lithofacies is chock full of invertebrate body fossils. So these were actually mystery fossils for the longest time. We found them throughout all of member B. Most of the time we just found this bottom chunk. We could not figure out what they were. We knew they were something biological because they were too regular, but we were just scratching our heads as to what the heck these things were. It turns out they're the carapaces of shed crayfish. And so we also have crayfish within this lake environment too, which makes sense. If we have a lake, crawdads like lakes, they like muddy environments. So we find their shed shells, and we also find their burrows as well. So this is another scavenger that was probably on the scene, tearing apart these dead frogs that were not in the microbial mats. A couple of plant impressions. So we get some really nice emergent vegetation that's preserved in these mats. We also get lots of ginkgo leaves. We get conifer bracts. And so there's a whole diverse flora. Obviously not the focus of this talk because we're talking frogs, hopefully, for the rest of this talk. But there are plants. So if we have any plant experts that are interested in this study, I'm more than happy to direct you out there to the field. So this slide's just showing the interbedded relationship. So here we have the microbialites, so the microbial mats, and laterally, not by that much, we transition into the laminated mud rocks. So these are really localized, isolated patches of these taphonomic modes. And then finally, the last way we preserve frog fossils in member B are these Tempestites. And so here we have mud rip-up class. So this is when a storm comes through, it rips up the mud in the bottom of the lake. We can get ripped up shells from clams and other invertebrates that are tumbled up within the storm. Think of a great big mountain lake, and a thunderstorm rolls across the lake, and the wind kicks up and it stirs up the bottom waters of that lake, and there's whitecaps out there and everything else. That's what these deposits are of. And they can also churn up frogs. And so this is a frog bone that's in the middle of a bunch of mud rip-up class. So this frog was deposited in probably one of those other two taphonomic modes and then got reworked by a thunderstorm. So since it's about dinner time, here we have a nice articulated frog leg all the way out to the toes, and a couple of frog legs up here. I'll tell you, I guess the first time I ever had frog legs was not in Louisiana. I was doing field work in Southwest Montana with my master's advisor, Dave Riccio, and we caught wind there was a Cajun restaurant north of Idaho Falls somewhere. So we drove for about an hour out in the sticks of the rolling hills of eastern Idaho. And sure enough, we found a random Cajun restaurant, and we dug into the frog legs. And because Dave's kind of a weirdo, he gathered up all of our frog legs at the end of the evening and took them back to his teaching lab. To this day, I still don't know what he did with them. But back to the matter at hand. All right, so we're still talking data and observations. So member C. So stratigraphically, we're moving up into younger rocks. We're definitely out of the Cretaceous. We're getting into the middle to the late Paleocene now. So the fauna in member C is a little bit different than member B. And we are about 10 million years younger now than we are in member B. So our fauna includes insect casings. So these are some modern caddisfly casings here at the bottom. And we do find fossilized insect casings or caddisfly casings in the Sheep Pass Formation. We do find tons and tons and tons of clamshells. We do find tons and tons and tons of ostracods as well, and we also get our frog bone beds. We have not found any plant fossils, and I've never personally seen snail fossils, but in the literature they are described. So we get two preservational modes of our bone beds. The first are articulated frogs laid out in single bedding planes. The second one are bone bed hash. And so these are actually entire stratigraphic layers, discrete layers that are nothing but frog hash. So this is actually the very first frog that Peter showed me. So this was his bird fossil that he took me back to his lab to look at. And you can see it's not very well preserved. All these little tiny gray, or I guess brownish dots, these are all ostracods. And then this is the left leg of the frog, the right leg of the frog, the hips, the shoulder, left arm, right arm, and then the head. And so these, where we went out and rediscovered where Peter found these things, and you pry up these big slabs of sandy limestone, you see in some cases anywhere from 1 to 5 frogs completely splayed out in these discrete bedding planes. This one, I'll give you a second to take a look at it. So Eorubeta is actually one of the largest fossil frog species that has ever been discovered. When Peter first showed me this sample, I thought that he had found a fossil jackrabbit because I saw this. I was like, oh, that's a knee, and that is huge. For those of you who've ever used a right in the rein, this is a right in the rein scale bar here. So this frog is longer than the scale bar. So this is the femur, this is the tibia here, this is one of the hip bones here, the middle hip bone, and then this is the head. So this great big wide semicircle is the head of this specimen. So it's not very well preserved at all. I wish we had found this thing about 10 years earlier before the elements hit it too bad, but this just goes to show how big this frog actually got. And Amy, when she did her study on the taxonomy of these frogs, she was fully expecting to find out we had several different frog species within the Sheep Pass Formation. But when she looked at this frog, looked at that previous frog, and looked at all the frogs through member B, they're all the same species for over 10 million years. All right, so the first taphonomic mode of member C, again, we're getting fully articulated frogs, kind of similar to member B. Just like a member B, we're looking at accumulation through time. There's little to no transport of these frogs, or else we would expect the bones to be disarticulated, kind of scattered throughout the landscape, but they're not. There are no signs of scavenging. We do have lots of ostracods, we have lots of bivalves, but for some reason, the ostracods are not eating these frogs like they were in member B. This is the second taphonomic mode. This kind of just looks like a rock, right? I guess they all kind of look like rocks, I'm told, but what's special about this one is every single one of these pink blobs is an individual frog bone. This entire slab is nothing but frog bones. And we have more than 3 horizons of these. So these are layers of frog hash. So we call them frog hash, but these are solid frog bone beds. They range in thickness between 3 to 5 centimeters, and they are irregularly bedded. The bones show no preferred orientation, and they're in bone-to-bone contact. It's just really weird taphonomy. So this is the actual bed of the frog hash. We can trace these things for over 1 kilometer, roughly north-south. So these were enormous frog die-offs. So you want to think of a stinky lake with a bunch of dead frogs on it? I mean, that's what this basin was when these frogs were out there rotting on the landscape. Just another image of the same. So again, each one of these pink blobs is an individual frog bone, all completely jumbled up. All right, so these frog bones in the bone beds show no signs of being exposed to the air, being exposed to ultraviolet radiation from the sun. All the bones are in a similar preservational state, which implies that maybe the animals all died at about the same time. There's no preferred alignment, so it doesn't look like a stream or anything like that was pushing the bones around. The bones are distributed erratically. The bones are found in direct contact with one another, which means that the flesh must have been gone. They must have been, all the stinky stuff must have been rotted off of them before they got removed. And so there's some modern analogs for how you can kill a bunch of frogs at the same time. So all these frogs that are belly up in this mountain lake up in the Sierras died from an early freeze. And so there's an early hard freeze. It killed all the frogs in the pond. So that's one mechanism. This is a bunch of frogs. This image is from Berkeley. This is a bunch of frogs that died recently from a fungal infection. And so right now, we're all familiar with the term pandemic. Amphibians have been dealing with a pandemic for years now, with the skin fungus that is spreading across the world. And so this is a frog die-off from that skin fungus. And so we can kill a bunch of frogs with disease as well. So what killed our kermits? Sadly, we can never tell. If you think of all the mechanisms to kill a frog, typically those are going to affect the soft tissue. And if we're left with just bones, it's going to be really hard for us to figure that out. But the fact that they are preserved can tell us a thing or two about the geography and the ecology of the area when they were alive. So this is an artist's depiction of what Utah, where you guys are at, or many of you are at, would have looked like during Eocene Paleogene time. So palm fronds, primitive mammals, so fuzzy things walking around, fuzzy things flying through the air, tropical to subtropical plants growing across the landscape. So what did Nevada look like during this period of time? What was the landscape these frogs were hopping around? So if we look at some frog CSI, so fully articulated frogs in member B and C, to me say that the frogs weren't bloated and weren't floating on the surface. Well, to think of a dead frog floating on a lake, things like birds are going to come pick at them, other scavengers are going to pick at them, fish should come pick at them. So to inhibit bloating and to have a fully articulated frog sink to the bottom of a lake, water temperatures have to be below 15 degrees Celsius to inhibit those bloat bacteria. If a frog is in a pond, just kind of floating around, and people have actually done studies on this, so fortunately not myself, but if you throw a dead frog in an aquarium and you watch how long it takes for it to fall apart, people have done these studies, it's about 3 weeks. So these frogs also have to settle to the bottom of the lake within 3 weeks of death. The microbial mats seem to have inhibited scavenging. And again, modern studies show that microbial mats build a natural sarcophagus around a dead frog and protect it from the elements. There are scavengers, ostracods, other invertebrates. Isolated bones seem to be from these storm deposits, since they're found also with beat up clamshells. What happens if your lake temperature is above 15 degrees Celsius? Let me take you to Southern California in the Salton Sea. So this is a big fish die-off in the Salton Sea. And because water temperatures are well over 15 degrees Celsius in the Salton Sea, for anybody who's ever been to the Salton Sea, it's just, you only have to go once, I would guess. You can see all the bloated dead fish that are kind of washing their way up onto the shore. The Salton Sea, up until probably just 10, 15 years ago, was actually the largest tilapia fishery in North America. So I'll leave that up to you, what you would like to have for dinner tonight. All right, so what does all this say about the geography then? What's the bigger story that these frogs are telling other than they survived the KP boundary, they're really primitive, they're huge, we find none of their tadpoles, so that's kind of weird also. But what does this say about the bigger picture of what's going on in this part of North America? So it tells us summer temperatures based upon stable isotope geochemistry. So summer temperatures of this lake basin hovered around 20 degrees Celsius. So at the hottest part of the summer, it was only 20 degrees Celsius. And that was based upon some work by Katie Snell at the University of Colorado when she was working on her dissertation. So how do you get a lake basin cool enough to inhibit bloating during a period of time where Utah is a tropical rainforest? So this is a famous graph from the paleoclimate literature. And we're looking down here in the Paleocene and the Eocene when global temperatures are 6 to 12 degrees warmer than they are today Celsius. So if the world is warmer, and how are we getting a cool lake basin in Nevada? This, again, the same latitude over in Utah is tropical. you shove the basin up in the air. And so as part of that big train wreck off the west coast of North America tectonically, our Shade Pass Basin, based upon Katie's work, the lake levels were somewhere between 2.2 to 3.5 kilometers elevation. So the lake was somewhere around 9,000 feet elevation. So the paleontology and the sedimentology argue that we have a very topographically high complex region where this lake basin was, where these froggies were living. Some more anecdotal evidence about why these frogs are preserved where they're at and how. So Steve Good was working on the mollusks, or the snails and the clams of this Sheep Pass Formation. And in his paper on this, he has an interesting anecdote where he was trying to find a modern analog for the snails and the clams that he finds in the Sheep Pass Basin. So again, paleontologically, this period of time in the Rocky Mountain region over in Utah, Colorado, and all the other laramide basins is more tropical. And so he looked to the tropics to find his analog for the snails and the clams. But there was no modern analog for the snails and the clams that he was finding. And so I guess he went home to the upper Midwest and was looking at snails and clams in Wisconsin and Michigan and Maine, and he found similar types of snails and clams. And so it was actually a temperate lake rather than a tropical or subtropical lake where he found his analog for the Sheep Pass invertebrates. So the vertebrate taphonomy, so how our frogs are preserved, suggests a cool temperate climate with storms rolling across this lake. So you can think of a landscape where you're sitting on the shores of this great big mountain lake, and big thunderheads making their way from west to east across this Nevada-Plano, stirring up all the stuff in the lake and dumping rain in this big carbonate lake basin. The invertebrate assemblage also suggests a cool temperate lake. The stable isotopes suggest high altitude. So all of these studies help shed light on the geography that Nevada, using multiple lines of evidence, was actually a high elevation environment during this period of time, which also helps support why it's so hard to find dinosaurs, why our dinosaur record in Nevada is so limited through most of the Mesozoic. Now, I've kind of set my career to trying to find those dinosaurs, which could be the subject of another talk someday. Maybe I'm just a glutton for punishment, but our rocks are really limited of that age, and so it's actually not that easy. So one final note, which keeps me up at night, scratching my head, it's so hard to preserve a frog. Think of a frog that you ran over in your driveway as a kid. It just kind of sits there sad, kind of squished and plopped out on the road. It's like, oh, that's sad. But think of how delicate that frog is. If we have a lake basin that can preserve a frog, then where's the fish? Where's the turtles? If it's a tropical environment in Utah, where are our crocodiles, bats, birds? If going back into the Cretaceous section, where's our dinosaurs? Where's our mammals? Where's our pterosaurs? So we have a really weird environment here where we're only preserving frogs. One of my mentors when I was in grad school said that he did some work up in Lake Titicaca in the Altiplano. And he said some of these lakes up in the Altiplano, they have one species of salamander and nothing else in the entire lake. Maybe that's what we have going on here. We preserve something like Lake Titicaca in Nevada. I don't know, but it's weird. I really want to find at least a fish. I want to at least find a bat or a bird, or pterosaur would be cool. But so the story continues on. And the most recent study on this, on the taphonomy, was just recently published in PeerJ. And if anybody's interested in that study, of course, I'm more than happy to share the link to that. It's open access. I'm happy to email that to you. Shoot me an email at the Nevada Science Center. I'm happy to send that paper to you. So a big thank you again to our collaborators, since this has been a huge team effort over a decade. Amy Henrique, again, at the Carnegie, Dick Hilton, Sierra College, Peter Druschka, formerly at ExxonMobil, Tina Campbell, for those of you who know Tina, Tom Madsen, he's the head of the Southern Nevada Grotto Association and works for the city of Henderson, great guy. Tyler, who many of you know, Frankie Bob Jackson came out for a field season. Amy's husband, Pat, at the Carnegie, great guy, was out there every field season lugging these big slabs of limestone off the mountain with our frogs. My fiance, Becky, at the Nevada Science Center also. My grandfather's band, the Duckwater Band of Western Shoshone, who let us camp in their front yard for a field season. And then the Bureau of Land Management for permission to collect all of our frogs. Our frogs, for those of you who are interested in follow-up studies, are reposited at the Carnegie Museum. the Sierra College Natural History Museum. And then for future studies that we're heading out to go do this coming summer, future specimens will go on to the Nevada State Museum in Carson City. So check out the Nevada Science Center. Again, we have free virtual programming. If you find yourself in Southern Nevada, you can check out our public research lab. We're on all the major social media platforms, and that's our contact information. If anybody has any questions after the presentation, I'm more than happy to send you our contact info, and thank you for having me tonight.
Thank you, Josh. That was great. Yeah. Lots of virtual clapping going on.
Yes.
Excellent talk. It's not so virtual. Anyway, we do have a couple of questions in chat. So
All right.
we'll ask those, and then we'll open it up to the those who want to just ask in person. So BJ is asking, do you get ash layers in these lake beds?
That's one of the very frustrating parts, is that this seems to be a volcanic hiatus in the Sierran Arc. So we don't get hardly anything that we date looks volcanic is detrital. We get no primary ash beds, unfortunately. And so that's why Peter had to get really creative with the dates that we did get. And so like doing the uranium, lead, helium dates on biogenic carbonates was really brand new at the time. So we had to really look at unorthodox dating techniques to get our dates. So that's a long, wordy answer for no, we didn't get any ash beds.
And Elaine, who I know also lives over in Arizona, but she's asking, have similar sites been found anywhere else in Nevada? And maybe expand that too.
Right. So this formation is found across east central Nevada. So for those of you who know the geography of Nevada, this unit's found all the way from just east of Ely, almost over to Wendover, it's been mapped, all the way over to Eureka, and as far south as the test site. But to my knowledge, this is the only section that has the prolific frog preservation. There's an occasional Eocene mammal that's found, but when we're talking about the Cretaceous and the Paleocene section, it's nothing but frogs.
And
All right.
seeing that there were only frogs were the only vertebrates in that environment, and you say invertebrates were feeding upon them, would it be safe to say that the invertebrates were the apex predators of that environment over the frogs?
It was definitely a buggy world, but I wouldn't say that they would be an apex predator, more of an apex scavenger. Frogs have no problem eating each other, so I would say the frogs are probably still apex predator. Frogs are just, everybody loves frogs, but the more you know about frogs, the less you'd like frogs.
Huh. Well, now that you mentioned cannibalism, if frogs have no problem eating each other, in those finds that you've had, have there been any signs of cannibalism between the frogs?
That's an excellent question. So as well preserved as these specimens are, there's been no gut contents discovered. When we published our taphonomy paper in PeerJ last year, one of the reviewers asked, well, why don't you send this thing off to the synchrotron to get it analyzed? Like, I'm funding this thing with my credit card. I'm not sending anything to the synchrotron. So maybe if somebody does a follow-up study and looks at the rare earth elements and stuff like that, like they're starting to do now, maybe there'll be some cool soft tissue stuff that you can't see with the naked eye. But right now, that's more than my credit limit.
Well, hopefully somebody they'll have the credit someday to do it.
Right. I'm totally open to working with people, or if somebody wants to take the ball and run with it, I'm happy with that. I think I've told my part of the story.
With so many bugs around, and I know that you mentioned that there hasn't been much finding of gut contents. Would it be fair to assume that, like frogs today, even though you're not sure what family of frogs these extinct frogs are related to, would it be a safe assumption that these extinct frogs did feed on some insects in their environment?
I would say that that's a safe assumption. I'd say if you look at the way we bracket lots of ancient behaviors is looking what the closest living relatives do, and all modern frogs are carnivorous, or at least insectivorous to some extent. So yeah, I would say that's a safe assumption that the frogs were eating the invertebrates, probably eating crayfish and other animals like that too. These caddisflies probably were tasty also.
I can probably imagine. And also with the complete specimens you've had, and even though I'm sure this might be kind of hard to analyze with their anatomy, but if you were to compare these frogs to present day frogs, when I'm sure we've all seen them in videos and in the wilds that they can hop and spring, would you think that these extinct frogs were quite as agile?
Yeah, even the big one that I showed, we call the Titan frog, that was the size of a jackrabbit. The leg proportions and the hip bones are all adapted to what modern frogs do. So it was definitely a hopper.
Okay. We have a couple of other people too that are looking. Hazel, I think you were next. I can't tell.
Yeah, thank you, Josh. That was great. I was curious about coprolites, and since we talked about that this morning, have any coprolites been identified in that mess of bones and whatever?
No, that's a great question. We have not found any coprolites, but that's not for lack of looking. We have found numerous invertebrate trace fossils, Peter did a number of thin sections and he found invertebrate coprolites, just peloids, but no vertebrate coprolites.
Okay. And Ian, I think you were next. You're in Garofalo.
Hello. Well, first off, great talk. It was very interesting. And
thank you.
Now, you described how this species of frog made it across the KPG boundary. And it's interesting because I know, I know amphibians have kind of been a subject of controversy regarding debates about the KPG extinction. And was there any significant amphibian extinction at the time that you know of, during that extinction event, or did amphibians get off pretty easily there?
So in researching this project, there was the quick answer is yes. Amphibians did take a hit at the end of the Cretaceous. But if you think of all of the devastating effects of, if the leading hypothesis is going to be a meteor hitting Mexico and that's going to be our extinction event cause, maybe there's a low altitude filter. And so if we have a delicate species that's living up at 9,000 feet elevation, maybe those environments weren't as impacted as lower elevation taxa. Now, how would we go about testing that hypothesis? Well, we need to find a lot more high elevation biotas, and sadly they don't exist. And so I'm arm waving.
Yeah, I know that's a persistent problem in paleontology, is we need a bigger sample size.
Right.
Yeah.
But yeah,
right.
Amphibians do take a hit, but for some reason these ones did fine.
Yeah. All right. Thank you.
You bet.
Yes. Yeah, it does surprise me, because I know I've heard and I've been told many times that bigger things they usually preserve easier than small things, which is why I know that a lot of adult specimens of a lot of animals are found more than juveniles. In which, with these frogs, have there been any juvenile frogs found, or have these all been adults?
So we have every stage. That's a great question. Thank you. We have found every stage of frog development, from recently metamorphosed to the titanophrog. But the only growth stage that we do not have are the larval stage. So we don't have any tadpoles, but we have every other stage in their development.
Hey, Josh, great talk.
Thank you, Jim.
I'm so glad you were able to come over and do this virtually. Anyway, one thing I find interesting is, we tried to do frog taxonomy in the past, in paleo, using the skeletons. And modern, if you look at poison dart frogs, there are hundreds of species. As I understand it, they're indistinguishable skeletally.
Yes.
Color patterns, songs are different. The time of year they mate, all kinds of things. But the skeletons are pretty conservative.
Right. So that's an excellent point, Dr. Kirkland. And we're talking purely morpho species here.
Yeah.
Martha?
Yeah, Amy.
Oh,
Oh, go ahead.
sorry. Martha, did you want to say something?
Yeah, my comment was, you mentioned them being in core samples, and we have a lot of at our core center Green River fossils that are in the core too, as well as. But since you're not finding other animals, my question is, in Utah at Secret Lake, which is up at Alta Ski Resort, it's like 10,000 feet. I think about the only species is a boreal toad, and one other amphibian. You might know, Gene, but could that kind of be and that's like a glacial tarn. It's a very high elevation glacial lake. Could that be a kind of modern analog to what you're seeing there? Since I don't think there's fish or other vertebrates or animals in Secret Lake, so
that sounds like a great analog. Since I grew up in Western Nevada, I was looking at mountain lakes in the Sierras for high elevation analogs, and even in the high elevation lakes of the Sierra, all the way up at the Sierra Crest, somehow catfish get up there. And so there's catfish up in these alpine lakes with the frogs. So I'll have to look into Secret Lake. That sounds interesting. But somehow fish just can get wherever the dang frogs are. So it still confuses me. But I'll look into Secret Lake. That sounds interesting. Thank you.
And if you haven't had a chance, Josh, do read some of the comments that are in the chat. Just people thanking you and saying great talk and stuff. It is. And, intrigued by the number of specimens, but the lack of diversity, it's very